Hello, and thank you to everyone around the world for joining us for this week's lab meeting. My name is Kristen Abood. I am the science editor at the Human Vaccines Project, and I will be your moderator today. More than 2 years into the pandemic, and with more than 5 million recorded deaths due to COVID-19, governments and public health experts are now shifting their focus to how we can live with SARS-CoV-2 long-term. At the same time, scientists are still learning more and more about how this coronavirus triggers disease and the diversity of immune responses that are mounted against it. Today, we will hear from 3 speakers who are part of a team that has collected the largest data set of T and B cell receptor sequences from COVID-19 patients on how the value of this data can lead to a better understanding of the clinical variability of COVID-19 and even help inform the design of vaccines and therapeutics. Before we get started with today's presentation, we'd like to congratulate the winners of the 2021 Michelson Prizes, who were selected from a global competition to receive $150,000 research grants to pursue work that will advance human immunology, vaccine discovery, and immunotherapy research across major global diseases. These 3 young investigators will receive their awards in a virtual ceremony to be held on March 10th at 11 a.m. Eastern Time, directly following that day's global COVID lab meeting. So mark your calendars for that, as we hope you will join us for both events that day. Now I am happy to introduce today's speakers, Dr. Felix Breden, Dr. Kira Neller, and Dr. Brian Corrie of the iReceptor project at the Simon Fraser University. Dr. Breden is Professor Emeritus at Simon Fraser University and is the founding chair of the Adaptive Immune Receptor Repertoire Community, a grassroots community of immunologists and experts in bioinformatics that was organized in 2014 to develop protocols and standards for sharing and comparing B and T cell receptor sequences. Dr. Neller is a data curator and bioinformatician with the iReceptor team, who works with researchers to curate COVID-19 adaptive immune receptor repertoire sequence data, as well as integration of single-cell data and analysis tools. Dr. Corrie is the technical director of the iReceptor project and has a background in computer science, particularly data analytics, data visualization, and bioinformatics. During today's presentation, please send me your questions using the Q&A function in Zoom. Feel free to use the chat for general comments, but please don't post questions there. Please submit all questions in the Q&A. I will ask our speakers a broad selection of your questions after their presentation, and we will have about 20 minutes for discussion. With that, it is my pleasure to now introduce Drs. Breden, Corrie, and Neller. Dr. Corrie, you can go ahead and share your screen.
Well, great. Well, thank you, Kristen, for the introduction and for the invitation to discuss our efforts at curating antibody B cell receptor sequences and T cell receptor sequences and how we've used those, or other people have used those to investigate the response, the immune response to COVID-19. So Brian, the next slide, you're going to run the slides, right? Yeah, okay. So yeah, in this talk, we'll have 3 sections. I'm going to introduce what we call the Adaptive Immune Receptor Repertoire Community. And this is a grassroots initiative to develop protocols for sharing these type of data. And mainly, I'm going to discuss why we feel we needed a new initiative for these type of data. And then Brian will talk about the AIRR Data Commons and the iReceptor Gateway, which queries this commons. And then Kira will discuss some latest results on applying this type of approach to the COVID-19 patient and vaccination data. So to introduce the AIRR community, we have to go over a little bit about the adaptive immune system, because again, you know, we've got a lot of people are developing genomic resources and integrating with health records. And, you know, why do we need a specific initiative for these type of data? And it really has to do with the adaptive immune system, characteristics of the adaptive immune system. So the adaptive immune receptor repertoire community focuses on the adaptive immune system, antibody B cell receptors and T cell receptors. And the adaptive system obviously evolves within the body in response to the pathogens that that individual has been infected with. And these type of data are critical to a huge number of any sort of diseases with an immune component. So vaccine development, but also autoimmune diseases, new cancer immunotherapies. And the basic point is that the adaptive immune system has to be just immensely diverse so that it can recognize any pathogens that the body may be infected with, presented with, and then any sort of new pathogens that we haven't evolved with, such as like the novel coronavirus. And then we have to recognize and not remove all the self proteins. So this diversity is the key to the adaptive immune system. And so these data really are difficult to share and compare for several reasons. First, this diversity, it's been estimated that there might be 10 to the 13th possible human B cell receptors in the whole human population. Any individual can have 10 to the 9th B cells and T cells. They're not all unique, but a large number of them can be unique. And then in any new study now, 10 to the 6th, 10 to the 7th sequences are sequenced per sample for before and after vaccination, within the tumor, outside the tumor. And so you start producing huge data sets. Here's some examples with one, the Burkitt lymphoma, 1.2 billion B cell receptors published in one paper. And these papers come out, at least once a week with these large data sets. So the diversity is one reason that it's so hard to share and compare these data. And then where does this diversity come from? Well, it comes from the process of VDJ recombination. And this recombination, it takes, as many people of this audience knows, takes these multiple V genes, D genes, and J genes and stitches them together, but it's on the germline in the chromosomes that this occurs. And this process is unique to these loci. So the immunoglobulin loci producing antibodies, the T cell receptor loci producing the expressed T cell receptors. This is a unique process to these loci in the whole eukaryote genome. And it's the first step, again, in producing the diversity of these receptors. And I like this slide because it has this little molecular scissors going in and snipping out the DNA between, in that case, a D and a J, and bringing that together, and then a V and another V and another D, and producing the recombined germline chromosome that then produces these receptors. And so clones, this kind of gives you a good definition of what a clone is. It's all the descendant cells from this original VDJ recombination and the original lymphocyte cell that's producing either T cell lineages or a B cell lineages. And again, these are unique. And so these processes are unique to adaptive immune system. And so this demands certain types of new databases where the germline genes are kept, and you can annotate the expressed gene against those germline genes, and then also unique bioinformatic tools to follow the clonal lineages and compare these. So next slide. And then this producing these data takes many, many steps. It's very complicated data, going from the germline genes to the expressed genes. And so these many steps have the different researchers can take slightly different approaches at all these steps. So you have to curate all the information on what tissues were used, how did they do genomic DNA or expressed RNA, how the bioinformatics was done, how the annotation was done. All these can lead to differences between approaches. So if we want to share these data, we have to curate all these different information on all these types of approaches that the researcher has taken. So that's another reason it's hard to share and compare these data. So in summary, they're very difficult. They're very complex and confidential. They require data on patient demography treatment, have unique bioinformatic tools. AIR-seq data possibly could be used for personal identification. So these data are hard to deal with, and that's why we started the Adaptive Immune Receptor Repertoire in 2015 to start looking at these data. I think we'll skip the next slide and then just start talking about the adaptive immune system, adaptive immune community. So it's a grassroots community. We believe in everybody who wants to participate is open to joining our working groups. And basically, we want to encourage and make it easy to share these data and to analyze these data in an interoperable way such that you don't have to recode the data every time you want to do a new analysis. And this ability really, you have a lot of people out there that have huge amounts of data on a small number of subjects, but by sharing these data across research labs, across institutions, you can increase the sample size, increase our ability to confirm trends within one data set by looking at other data sets. And next slide, please. And really the working groups are the key to the adaptive immune receptor community. We have multiple working groups. They meet once or twice a month. They've been meeting for several years. And this is where we produce the standards and the protocols that we're trying to develop from a community approach and put out there for the full community. And next slide, please. And this emphasizes the real way the work is done and how the working groups are really the key to the adaptive AIR community. One of our first papers was from the minimal standards working groups, and it was published in Nature Immunology. We call it MyAIR, the minimal metadata standard for depositing or curating these type of data representation, starting with, how can it be represented in a computer database. The Common Repository Group has developed the AIR Data Commons, which Brian will talk more about. But kind of that one of the key points is that these working groups are open to anybody who wants to work with us. And then we publish these standards and protocols as a scientific publication that has to be approved by the full AIR community. So we try to build in community support and buy-in by making it open to the community and approve all of these by the full community. So we'd be happy if anybody wanted to work with us, and there's the AIR community website. And we've got a AIR community meeting number 6 in La Jolla in May, and hopefully that will be a hybrid. It would be a hybrid meeting, but we'll have a large component of face-to-face. And we definitely encourage anybody who's interested in this to contact us and to join the group and to come to the meeting. And I'm now going to pass it on to Brian for the more details of how we do all this. Thank you.
Thank you, Felix. Yeah, so I'm going to talk about the iReceptor platform in a bit more detail. So I'm a computer scientist, and I'm kind of the technical architecture of the platform. So I'm going to go over a fair bit of stuff fairly quickly, touch on a lot of different things, and then hopefully that will give us some food for discussion later on in the Q&A. So Felix has already talked about this, but I'll just reiterate a couple of things. So one of the big drivers for the AIR Data Commons is this data deluge that we're seeing from the next-gen sequencing. And I think the key thing here is that there are many subjects per study, many studies per labs, and hundreds of labs that are producing this data. I'm going to talk about how much data is in that data commons in a bit, but it's really only scratching the surface. And so that's kind of one of the big driving factors. So the philosophy behind the AIR Data Commons is that we're having a distributed set of repositories allows for things to scale up. So we can have billions of sequences across many studies because we can have hundreds of repositories. One of the things that Felix mentioned is that one of the key things that we want to enable is for researchers to curate and manage their own data. So have data stewards curate data at their home institutions, but still be able to share that data very easily. And one of the things that we've developed, I'm not going to talk too much about in this talk, is we developed a database platform that you can download to use to store such data and participate in the AIR Data Commons. So really what we're trying to solve is meet the researcher needs to basically ensure that AirSeq data is fair. So people probably have heard of the FAIR principles: findable, accessible, interoperable, and reusable. So we want to ensure that researchers can find AirSeq data and federate that data so that they can reuse it. And really the reuse is about deriving new insights from data that has already been produced. So that's kind of one of the driving factors for the iReceptor platform in the AIR Data Commons. So how does iReceptor work? So there's kind of two main components. There's the iReceptor Scientific Gateway and the AIR Data Commons. So the AIR Data Commons is this distributed set of repositories. I'm going to talk about in detail. There's currently 5. There's several coming online in the near future, and we expect the number of repositories to grow, and I'll talk a bit about that in a second. The iReceptor Scientific Gateway is essentially an interactive web-based data discovery, exploration, and analytics platform. And so that allows you to explore the AIR Data Commons. So how and why does this work? Well, the only reason that this is really possible is because of the standards that have been developed by the AIR community. So this is an international body that has an extremely large amount of buy-in from a pretty broad spectrum of the research community. And so these standards are fairly widely adopted. What standards like this mean is that a researcher can send a query to any one of these repositories. That query can be sent to any of the repositories in the AIR Data Commons, and that query is identical, right? So it's because it's standards-based, the query is the same to all repositories. And probably more importantly, the data that comes back from those repositories is also in the same format. And so that makes it easy to federate that data and combine it so that it can be brought together and presented to the user. So really what the iReceptor Scientific Gateway is doing is it's hiding the complexity of sending queries out to many, many repositories, recombining that data and presenting that data to the user so it's easy for the user to use. So how does this work? So basically the iReceptor Gateway searches the AIR Data Commons. There are currently 5 repositories in the AIR Data Commons, 68 studies, and over 5 billion sequence annotations. So there's sequence annotations. So these aren't the raw sequences that we're talking about. These are the annotated sequences that have been run through the pipeline to do the VDJ annotation. So these are highly valuable data. These are not the raw sequences that you get from SRA. In addition to that, they're basically covered, annotated with metadata about the studies, the subjects, and samples. So again, highly valuable data in terms of trying to understand where the data came from. So there's 2 workflows that we support basically. One is exploring through the Repertoire metadata. So this is basically the workflow that is most commonly used. Researchers are trying to explore the metadata. I'm going to talk about this in a second, and try to get an understanding of and find data of interest to them. So specific disease, specific age ranges for subjects and samples, et cetera, et cetera. The other workflow that we support is basically doing, kind of think of it as a Google search for the AIR Data Commons, where you just basically type in a string of interest. Typically, these are CDR3s, which are very important. Basically, the kind of defining feature for a clone, which is kind of what Felix talked about a bit earlier. So those are the 2 workflows. I'm going to talk about the metadata workflow for starters, and basically use finding COVID-19 data as an example. So Felix mentioned the AIRR standards. So the AIRR standard basically has over 80 metadata fields. Now, these fields are not required. The AIRR standard can be best thought of as a recommendation to people who are performing studies as to what you might think about as being valuable to capture. So not all studies need to capture all aspects of these 80 fields, but the more the better. And so basically what the iReceptor Gateway is doing is making it easy to search over these fields and finding data of interest. So just a really quick example, in order to find COVID-19 data, there's basically two mechanisms that you can use, either a text search, which is basically looking for keywords, or you can actually look for kind of ontology terms around disease diagnosis, for example, COVID-19 as a specific disease from a specific disease ontology. Once you search and find those, type in that search, you basically get a set of filtered studies that meet your search criteria. So in this case, there's 1 billion sequences from 3,531 repertoires. That data was found in 3 remote repositories, and it comes from 20 different studies. And you can see kind of the pie charts that kind of give you a summary of that. Once you find data, you can kind of drill down into some summary statistics about each of the repertoires. So we have a set of repertoire stats that you can do. And these are giving you things like V gene, D gene, J gene distribution. So kind of looking at the distributions of the genes that are part of the specific repertoire or sample, and junction length. And if you want to drill down into a specific gene, you can go down to the gene level and down to the allele level as well. So this is basically kind of that exploratory data analytics that you can do to try to help determine whether this data is of interest to you as a researcher. So the iReceptor gateway is kind of fundamentally about data discovery at this point anyway. And so once you find data, of course, what you want to do is you want to download that data for reuse and further analysis. So if you happen to have gone through the workflow where you're looking at metadata and you found a study that you find interesting, at the click of a button, you can kind of download that data and perform offline analysis, downloading a whole study in this case. And there's a whole range of types of searches and filters and sequence of workflows that you can go through. The second one that I'm kind of pointing out here, this is probably, I actually don't even, yes, it looks like somebody did a search for a download for a CDR3. So they're interested in a specific, probably clone that was heavily expanded, and they want to find out if it's found elsewhere in the repository. And it was only found in 301 sequences across the entire 5 billion. So that is obviously not a very common CDR3. And so, but if you're interested, that's critical to your research, it's quite easy to download that. So I'm now going to jump off, stop talking about the gateway and the platform and talk a little bit more about the AIR Data Commons and the AIR Data Commons use. And I'm going to drive this around COVID-19. So the iReceptor gateway and the AIR Data Commons has been around for some time. Initially, there were 2 repositories, the iReceptor Public Archive, which is the repository that we run here in Canada, and our collaborators at the University of Texas Southwestern Medical School, VDJ Server, that's Lindsay Cowell's group in the US. And as you can see over time, there's a large number of small step increases that we added studies to the AIR Data Commons. You'll see this rather large spike in increase in data around June of 2020. So that's that orange block is the COVID-19 repository that we created. So we spent a fair bit of time curating COVID-19 data in the early days. I'm going to talk a little bit more about that in a second. And one of the really exciting things for us is that we have, so VDJ Server and iReceptor were the key repositories, but we have a set of new international repositories that are either online now or coming online. So we've got VDJ Base, which is a repository from Bar-Ilan, Israel. We've got iReceptor, which is from DKFZ in Germany. We have a repository from Sorbonne. We've kind of taken some liberties and we kind of show it as being active now, but it's not quite. And there's a group at NICD in South Africa, Catherine Schieper's group, that is actually about to come online as well. So not only is the data growing, but the number of repositories are growing. And I think that's an indication that the approach we're taking is an attractive one. Oh yes, last comment is, so VDJ Server works slightly different than we do. We do a little bit of ad studies at a time, whereas VDJ Server does these large kind of step increases in there. So they load many studies at one time. So that's why we have that big step increase. So we have over 5 billion rearrangements in the AIR Data Commons today. So I'm going to talk a little bit about how COVID drove our data sharing in your data commons. So the first preprint or paper that we were aware of is the Nielsen et al. Paper. It was published as a preprint in June. And essentially about 3 weeks after that paper was published and the data was available in SRA, we were able to run that through a pipeline, annotate the data, curate the metadata. And so we had a really quick turnaround. One of the key things is that so these papers came pretty fast and furious. So there are another 4 papers, Galson et al., Minervina et al., Schultes et al., and Liao et al. Basically were published as preprints in the next several weeks. And we worked quite closely with those groups. And I'm going to talk about the Schultes paper in a little bit more detail, because I think COVID-19 really changed the way researchers were sharing data. And in particular, the binder group, National Binders Group and the Schultes paper, they actually reached out to us before the paper was released in preprint, and the data was in the Airdata Commons before the preprint paper was released. And in particular, iReceptor was cited as a source for that annotated data in the actual paper itself. And this caused, I think, quite a stir and quite a significant increase in use, which I'll talk about in a second for the Airdata Commons. Another interesting thing, this is an incredibly rich data set. It had 46 subjects, had both IG and T-cell data, 15 million annotations. And the most interesting thing, I think, and why people thought this was really important, it was the first study that had time series data. And it was quite a rich time series data set. So they had up to 9 time points for a single subject and different time points out to 55 days past symptoms onset for that data set. So that was a pretty key turning point for the use of the Airdata Commons. So there are lots of papers that came out after that. We worked closely with the researchers to publish them. Adaptive Biotech published a big T-cell data set. That was added to the repositories. Papers continued in 2021. And Kira is going to talk a little bit more about some of the vaccine papers, but these are the 4 vaccine papers that we've been working with. And one of the interesting things about this is that we work closely with both. So these are papers from 2 groups, 2 papers from 2 groups each, the Elibeti group and the John Wary's group. And we work closely with Julian Zhu from the Elibeti group and Aaron Rosenfeld from the Wary group to curate these data sets so that we have a bit of a pipeline with these researchers. So when new data comes online, it's very, very simple for us to get it up into the data commons. And so this curation continues. So really quickly about use, in kind of June 2020, when that binder Schultes paper came online, we went from having kind of 10 to 15 inquiries about usage to 70 users requesting accounts on the gateway. And this pie chart is basically showing the usage, the number of users added by region since June 2020. And that data is basically showing that there's a pretty broad North American, Europe, and Asian contingent to users of the community. But I think interestingly, 18% of the users or 72 users from industry have requested accounts on the iReceptor gateway. So I'm going to skip over this fairly quickly. I've got about 5 minutes before I'm going to hand over to Kira. So there's just a couple of things that we've added for the kind of the future of where we're going with iReceptor. So one of the things that Felix mentioned is looking for clones. CDR3s are kind of the definition for clones, and it's quite easy to search for CDR3s. This is that other workflow. And what typically people are interested in is looking for public CDR3s. So CDR3s that are found across many subjects, across many studies. But in the Airdata Commons, we do not have any information about what binding characteristics those CDR3s have and what epitopes or antigens they bind to. People may know of IEDB. It's the International Epitope Database run by Bjorn Peters and his group. They have an enormous amount of information. So We've basically integrated IEDB queries into the iReceptor platform. So if you search for a CDR3 in the AIR Data Commons, we also search to see if there's any known antigen specificity to that CDR3, and then we can send you off to IEDB to look into that for more details. We're also doing some work in combining AirSeq and single-cell immune profiling. So bulk AirSeq, as we've been talking about it, is very, very deep across many, many cells. So you get millions of sequences. Single-cell immune profiling is much rich in terms of the information it provides about each of the kind of receptors, paired chains, a better clone resolution, and gene expression for the cells. But typically you get a small number of cells per sample. So these two techniques are complementary. And so typically in the AIR Data Commons today, you get a look at kind of what the AirSeq clones are and the VDJ gene combinations and things like that. In the single cell world, you get much more information about what the gene expression signature is. And so what we're working on is curating both AirSeq and single cell gene expression data in the AIR Data Commons. So the AIR community is working on a standard. So we've been working with 10x. So this is a little bit, this slide is a bit 10x focused, but it's general from a single cell perspective. 10x recently, in their recent software release, released the ability to produce AIR compliant repositories. And we have a number of 10x immune profile studies in the repository. The AIR community is working on a single cell gene extension in collaboration with 10x, and hopefully we should be able to load single cell and gene expression data into the AIR Data Commons repositories. iReceptor is working on this, so we have a software stack for implementing a repository. So if you wanted to download the iReceptor turnkey, you can do that and install it at your own site. We hope to be in production with that in mid-2022, so June timeframe, where you'll be able to actually curate single cell data and do single cell user workflows. Another thing that we're actively working on. So today, basically what you can do is you can select data in the AIR Data Commons through the iReceptor gateway, and then you download it and perform offline analysis. And tomorrow, what we're moving towards is being able to choose the analysis that you want to do, run an analysis job, with that data never really used, the user and the data never really leaving the platform, right? So you don't have to download anything to your desktop. You don't have to install the software on your desktop or on your local computers. And then you can get a bunch of different types of analysis results through the platform without ever leaving the platform. And the last kind of extension that I'm going to talk about is just talk about iReceptor, the AIR Data Commons, and machine learning. So machine learning is a pretty active area of research in understanding the immune response. And the AIR Data Commons is important in that domain because of the rich data system, data ecosystem that the AIR Data Commons is. Because we have such rich metadata and consistent standards compliant rich metadata, it's fairly easy to use that data to train machine language classifiers. So I say here, there's 5 billion reasons to use the AIR Data Commons because there's 5 billion rearrangements that you can use, to each of them with extensive metadata to use to train your machine language classifiers. And then of course, the hope is to have machine language classifiers to better understand the immune response. One of our collaborators is from the University of Oslo. They've developed ImmuneML, which is an open source machine language platform for AirSeq data. It uses Galaxy, if people are familiar with it, as a computational platform. And they have classifiers for immune repertoires and BCRs and TCRs, and B cell receptors and T cell receptors. One of the things we've done in collaboration with the ImmuneML group is we've actually integrated that into the iReceptor gateway. So when you do a download, or when you have found a data set that you can download, rather than download it, you can send it to Galaxy, the ImmuneML Galaxy. And basically that allows you to kind of transfer data that you find in the AIR Data Commons into the ImmuneML Galaxy platform and use that to either train ML models using the Galaxy platform, or do data analysis using machine language models that you've already got trained. And hopefully in the future, we want to move those ImmuneML classifiers into analysis apps on the iReceptor gateway itself. And on that note, I'll pass it over to Kara.
Great, thanks, Brian. So as Brian has described, the iReceptor team has been working really hard to, stay on top of the COVID literature and curate these studies. So in my part here, I'm going to be talking about really the value of having all this data in one place in terms of sort of data reanalysis, as well as some of the key insights from these studies. So the data curation efforts from the iReceptor team really have been making an impact in terms of the research community and the research that's coming out. So an indicator of that, of course, is how many times iReceptor has been cited. And we've been cited 32 times since June 2020. And this is either as the location of deposited AIR-seq data, so in the data availability statement, the location where published AIR-seq data was found for reuse. So here, driving new insights from data that was available in the AIR Data Commons. And finally, as a general resource for finding AIR-seq data. So here, I'm just going to provide a couple examples of some cases of COVID-19 data reuse, really leveraging the AIR Data Commons. So Meisman et al. Integrated longitudinal data from 2 studies, and they found that there was increasing TCR clonal diversity with time and disease severity in COVID patients. And Hemming et al. Integrated 7 studies using the AIR Data Commons, and they were interested in what they termed neuro-COVID, or the neuroinflammatory symptoms that are seen mainly in the severe COVID cases. And they found that this neuro-COVID was associated with T cells that had an exhausted phenotype, as well as broad clonal T cell expansion. Next slide, please. So if we take a look at these studies, particularly the early studies, we can get some immune repertoire-based insights from them. And just before I go on, I'll mention that the majority of these studies are indeed in the AIR Data Commons, so you can explore them in more detail there. But we found that many studies were reporting that in COVID patients, the adaptive response was mediated by what we've talked about, those convergent clonotypes or public clones. So in fact, this response, or these expanded clones, were so indicative of COVID infection that the FDA approved a TCR repertoire-based test for prior COVID infection. So this is the T-Detect, and it's produced by Adaptive Biotechnologies. And I'll just note here that the public clones are very important also for things like vaccine leads. And in COVID patients, studies also reported that despite the many potential V genes that can be used for VDJ recombination, there were reports of biased V gene usage in these responding clones in COVID patients. So there was reports that IgH V353 and V366 were greatly increased in anti-SARS-CoV-2 antibodies. So just sort of bringing this back to what Brian was mentioning before about the different apps that will be available in coming days, is so you could do this type of analysis easily on the gateway through the statistics app to look for gene usage. And finally, there were some interesting results that came from healthy subjects, and where it was found that there was actually some evidence of pre-existing SARS-CoV-2 immunity, and this was thought to be due to exposure from other beta coronaviruses, so those associated with sort of common cold variants. And this was evidenced by precursor neutralizing antibodies in healthy subjects, and as well SARS-CoV-2 induced T cells from COVID patients were found to be cross-reactive to these common cold beta coronaviruses. So the advantage really of having all of this COVID AIR-seq data in one spot in the AIR Data Commons is that we can really verify some of these trends on a larger scale. So that's what we did for this one case, where we wanted to see a public clone that was reported in the particular study. Here's Shomurodova et al., we wanted to see how public was it really making use of data that was in the AIR data commons. So Shomurodova et al. Reported in their study that there were 16 out of 17 COVID patients with a specific HLA allele. They reacted to a single spike protein epitope, so they termed this wild Q. And 1 out of 24 healthy donors reacted. So the wild Q specific TCRs had a conserved CDR3 motif, which I've indicated here. And if you take a look at it, it can actually be decomposed into 24 possible sequences. So what we can answer using the ADC API, and what we did answer, as I'll show on the next slide, is have others reported this motif, and how prevalent is it in COVID repertoires versus healthy repertoires? So here I'm showing the results of our analysis. So what we did was we started out with COVID repertoires. So we were looking specifically at the TRB repertoires. So we gathered all the COVID TRB repertoires available in the AIR data commons. So there was 1,616. And we looked at the healthy ones, of which there were 38 repertoires. Now, interestingly, although there's more COVID repertoires than for healthy, the average sequences per repertoire in the COVID case is much less, so an order of magnitude less than in the healthy repertoires. So when we actually searched for repertoires with this motif, we found the motif was present in 87% of the COVID repertoires, and interestingly, 74% of the healthy repertoires. So we actually found in our sort of reanalysis case that there was more that this motif was more public in healthy repertoires than was previously reported. However, our results do agree with the fact that this is a COVID sort of that the COVID expansion of this TCR clone, because if you look at the average motif frequency per repertoire, you see over an 11 times increase in the COVID case. Next, thanks. So the conclusion sort of summarize what I just described. So we found that the motif is indeed present in both COVID and healthy TRB repertoires. It's much higher in COVID than in healthy. And the fact that we were able to combine, or effectively increase, the sequencing depth by looking at all of those healthy repertoires, we were able to really drill in on this COVID-specific expansion. So with that, I will sort of switch gears and talk about some of the repertoire insights based off of recent SARS-CoV-2 vaccine studies. And I'll just say that the majority of the studies I'm going to be discussing, especially the ones with the green sort of check marks as I move through, are ones that are available in the AIR data commons, queryable through the iReceptor gateway. So when we think about the SARS-CoV-2 vaccines, and I'll note that all the studies in terms of the repertoire-based studies have so far only been looking at mRNA vaccines. The hot topics in this field really are how durable is the vaccine? So how long is it effective? How do vaccine-induced responses differ between SARS-CoV-2 naive and previously infected individuals? And of course, what's the impact of boosters in these two groups? How effective are available vaccines against variants of concern? And finally, we know how variable the adaptive immune system is between individuals thanks to the VDJ recombination. So how does this heterogeneity between individuals affect the vaccine response? And I'm going to be focusing for this talk just on questions 2 and 3. So for the question of how do these vaccine-induced responses differ between the naive and previously infected individuals, Joel et al. First tackled this question. And what they did here was they had a cohort that was composed of both SARS-CoV-2 naive and recovered individuals, and they sampled the BCR repertoire at 4 different time points. So time point 1, primary vaccination, and then there was a pre-boost, a boost, so the second vaccination, and a post-boost. And interestingly, what they found was that in recovered individuals, so those who had prior exposure to SARS-CoV-2, they found antibody and memory B cell responses were significantly boosted only after the first dose, not the second. So the standard sort of biomarkers of circulating antibodies, neutralizing titers, antigen-specific memory B cells, they found no significant increase in this population after the second dose. Next slide, please. And then in October, the same group again was sort of investigating this question with the same cohort, but now they're looking more into the durable immune memory and more specifically, the question of how these vaccines fare against variants of concern. So what they found was that regardless of the prior infection, the vaccine-induced memory B cell clones were similar. So if you look on the heat map on the right there, this is just showing the gene usage profile of these responding clones. In the blue or red is showing whether they've had prior COVID exposure or not. And then the binding actually is, well, I'll get into in a second, but so what we can say sort of from the first statement was that due to the similar IGHV gene profiles in terms of the blue and the red columns, so infected or not infected, these were similar. So it suggests that they're recruiting the same clones. However, the gene usage, sorry, if I could go back, gene usage actually clustered based off of the antigen specificity to the variants. So if you look at sort of the purple and the green, the purple are they are clones that were able to bind the spike without the RBD. Light green is ones that can bind the wild type RBD, and then RBD++, dark green are ones that could bind across variants. So really what we're seeing is that the clustering occurred across variants. And if you look sort of at the middle panel, their results showed that there was increased usage of VH353 and 366 in these RBD cross-binding clones. Next. So that study that I just discussed sort of leads in, of course, to the question of how effective are these available vaccines against variants of concern. And Schmitz et al., they characterized a vaccine-induced public antibody, so they termed this 2C08, and they found that it was protective against SARS-CoV-2 as well as emerging variants in a mouse model that they used. And they searched for these 2C08-like clonotypes that were found to respond in SARS-CoV-2 infection and vaccination, and they found them both within their study cohort as well as published sequences from the literature that others had reported. So they found the 2C08-like clonotypes in 41 out of 181 individuals total. And I already sort of described those cross-binding variants from Joel et al. And they found that with vaccination, there was more variant cross-binding memory B cells versus mild infection alone. And if you look at sort of the overlap of those clones that could bind both the wild type RBD as well as the RBD of different variants, they found that the variant binding memory B cells were more hypermutated than wild type only binding cells. So this suggests that they've gone through a sort of affinity maturation, that they're more going to have a more sort of potent, effective response. So just before I wrap up and sort of pass things back to Felix, I just want to reiterate that all of these studies that I've mentioned are available in the Airdata Commons, and we really invite everybody to sort of use them through and query them through the iReceptor gateway and drive new insights through reanalysis. So now back over to Felix.
Great. Well, thanks, Kira. We'll have a little time for questions, but we do want to just kind of summarize what the opportunities and challenges are for the Airdata Commons, what we're hoping to work on with the human vaccine project. really try to achieve this network effect. So we're starting to grow, but we really, anybody who wants to share their data through the AIR Data Commons and establish a repository at their own institution or place it in one of the repositories should contact us. We really want to extend this new sharing culture that's really been spurred by COVID-19, but all of the different diseases that are, immune therapy, cancer immune therapy, autoimmune therapy, sharing these data is very important for all of these types of disease research. So we're hoping to expand that culture of sharing. Certainly AIR-seq data, B-cell and T-cell receptors, they have to, they can't be analyzed alone, and we're bringing in the other data types, like Brian said, with the single cell approaches, but bringing in the gene expression and proteomics, and hopefully bring in electronic medical records. And one way that this is moving ahead, trying to integrate all of these different types of data, but take this AIR community grassroots approach, is through a big data immunology community, a committee of the International Union of Immunological Societies, with the idea that, getting people to adopt these shared standards so that it's easy to share these data and it's easy to do interoperable analysis. It's the funding agencies can help, the journals can help, but perhaps the societies, the immunological societies, are going to be one of the ways to really push acceptance of these types of data. And so anybody who wants to help participate with us, again, can join the AIR community, or in the next slide, we have here it is, support@iReceptor.org, in terms of help sharing these data, setting up repositories, getting your data into the AIR Data Commons. So with that, we have a slide acknowledging our colleagues and collaborators such as VDJ Server, 10x and Genomics, and MedGenome in our iReceptor Plus project, and the funders for the Canadian. But we could turn to questions, I guess.
Wonderful. Well, thank you very much for a very clear presentation from all three of you. So we'll jump right in. So that we have first of all, some compliments on the platform. So kudos to that. And a question on, is it possible to search for T-cell or B-cell epitopes?
Right, yeah, somehow I lost that question, but I was excited to see that question. I'll answer it a little bit, and most of these, it'll probably then go to Brian, who's doing all the technical work. But one thing we're trying to, we've got several different initiatives to try to integrate, really from the germline gene inference, from the genotype all the way to the phenotype, in terms of going through starting with the germline genotypes into the naive repertoires, into the antigen stimulated repertoires, and then for particular CDR3s or particularly expanded clones, then looking at the binding characteristics. And so I know one thing we're developing is where when you search for a CDR3, then you immediately learn what data might be in the IEDB database for CDR3s, but for that CDR3. But I think there's other initiatives too that Brian might mention.
Yeah, just really quickly. I mean, I think, we're working on that kind of epitope antigen kind of connection, and we're doing that with IEDB, but it's kind of one way right now. If you search for a CDR3 in the AIR Data Commons, we can link you to antigen specificity and epitopes through IEDB by sending you to IEDB, but you can't search for CDR3s that have binding characteristics to a specific antigen in the AIR Data Commons directly. So you kind of have to go back and forth between IEDB and iReceptor. Just so happens that we have a project proposal out right now to solve that problem. We hope it all gets funded.
Okay, very good.
An NIH resource grant, U24, with the full AIR community. Yeah.
So Brian, this one might be for you as well. The question is it possible to search iReceptor for specific VH genes rather than the CDR3 sequences?
So that's completely possible. And through the gateway, you can do that interactively. So most of these searches, like a CDR3 search, we worked really hard to make those fast. So you can search all 5 billion records in, I don't know, a minute and 30 seconds. And you can do the same for VDJ genes. You can search for specific junction lengths or CDR3 lengths. What's challenging is to do kind of search for both a combined V and J So if you ask for a V and a J, that's actually quite a complicated search. And our gateway is targeted at being interactive. So we kind of terminate any query that takes more than 3 minutes. It can be done through the Airdata Commons API, but the gateway kind of has this limit of 3-minute queries. So if it's going to take longer than 3 minutes, if you when I flip through the analysis page, there is actually a heat map there that was actually a VJ gene usage heat map. Okay. So that's possible as analyses, but not through the interactive workflows.
Right. Okay, so we have 2 questions coming in on the HLA type associated with the TCR information. And can certain TCRs be mapped to a group of individuals based on their HLA genotype?
Probably Brian.
Yeah. So one of the extensions that I actually didn't mention in my extension slide, but that Felix just alluded to, was we are working on, so there is an AIRR community standards extension that actually is recording genotype, both kind of the VDJ genotypes, but also the HLA MHC genotype. And so although we don't have much data and the standard is new, the AIRR standard, and therefore the repositories have the capability to do it, we just don't have much data around that at the moment. So it's a new thing, certainly something that we've got on the radar.
Okay, right. And so this one might be a question for you, Kira, which is, can this data be used to discover and develop idiotypic vaccines against COVID?
Unfortunately, I'm not sure what even that type of vaccine is. I'm not not a vaccine expert.
Right. Okay. Well, maybe I'll go back to them and ask for a little more for the question. We have another question, which is about information specifically related to regulatory T cell receptors. Is there any specific information regarding that?
I mean, we certainly have that sort of cell subset curated within the Airdata Commons. Off the top of my head, I can't recall if any of these COVID-based studies sort of looked at that cell subset specifically. It's interesting, actually. A lot of the TCR repertoire insights in these vaccine studies are really just emerging. So I saw just sort of December, January is when there were a few papers based off of how the TCR repertoire contributes to vaccine response.
Right.
I urge the questioner to sort of investigate those recent papers.
Okay, great.
Yeah, if I could, I mean, these questions are really exciting, and I think they really show how what we need is to, again, kind of expand this culture of sharing where, you know, these population-specific questions or the different cell types, you know, and the more data we get into the Airdata Commons, then it's easier to answer these questions. And of course, you know, again, it can help support people that are interested in uploading into one of the public repositories, or in establishing their own turnkey repository by downloading that software that's open source, and we can help teams that want to develop their own individual repository.
Okay, right. And so, Felix, on that line, we had another compliment to the platform, and also a question on, you know, that you pointed out that this data can be sensitive, and how do you address issues of individual privacy or identification in terms of?
Well, right now, the Airdata Commons only curates data that's already been out in the public domain. But it might be an SRA and not very usable, but then we work with the authors and put it into a much more usable form. As part of the iReceptor Plus project, a large EU Horizon 2020 project, we're developing some security ability to keep you know, more secure data, but that's still being developed. And
well, let me just say thank you again to all 3 of you for today's excellent presentation and this work. And I think we all hope that one of the lasting legacies of COVID will be more data sharing and
absolutely,
usage. So Thank you so much. And also for taking the time to answer the questions. And as always, I'd love to thank today's attendees for participating in the webinar and submitting such great questions for our speakers. We continue to be so fortunate to have such an engaged audience for the lab meetings. And I invite everyone to join us 2 weeks from today on February 10th. Our speaker, I'm sorry, it's not February 10th, but 2 weeks from today, our speaker that day will be Dr. Salim Abdul Karim, Director of the Center for the AIDS Program of Research in South Africa and a Professor of Global Health at Columbia University in New York. He will be presenting on COVID-19 in South Africa, an update on Omicron. And if you're interested in more research on COVID-19, please sign up for the HVP COVID report. It's a bi-weekly newsletter that provides insights from experts around the globe and highlights the latest scientific articles and data. Finally, please visit our website and follow us on LinkedIn, where the full webinar series is available, including a recording of today's. And with that, I'll say thank you again, everyone, for participating. Stay safe, and we hope to see you again in 2 weeks on the next Global COVID Lab meeting. Thank you so much.
Thanks again.
Thank you.
Bye.